Bismillah ar-Rahman ar-Rahim. Now actually we are dealing with the serial of lectures on infectiology, that is infection and the disease. And I was talking previously with some of the gram-positive organisms. Now I have reached the Clostridial infections. In Clostridia, they are the gram-positive organisms. Basically, they are gram-positive and they are rod-shaped, and as per clinical importance is concerned, they are divided into four main diseases actually. There is the Clostridium, what is called tetanus. It is Clostridium, tetanus and botulism. Clostridium botulism, Clostridium tetanus, Clostridium Perfinges, and the Clostridium difficile. These are the four important organisms related with the genus. I would say this is Clostridium genus, and it's got a variety of the species, and there are more than 100 species described. They are a saprophytic organism. They are present in the environment, in the soil, elsewhere. And their culture media, they like to involve the organic matter and putrefying matter, I will say. They are prone to develop the best media. They like or prone to develop in the media which contain the organic material rather than inorganic material. And I am Professor Syed Ali Hazar. I am representing the Genus and Medical University, Karachi, as Senior Visiting Professor of Internal Medicine. So today I want to talk the third part of it, probably, and this is the Clostridium balchi. At one time it was called Clostridium balchi, now it is called as Clostridium Perfinges. In our days we call it Clostridium balchi. But this organism is important as per medical and allied is concerned, and surgical and allied is concerned, all subjects, including the gynecology. Now this organism, Clostridium Perfinges, is important. It's gram-negative organisms, you see, and they are the same one as I previously told. They are violet color, and they are therefore gram-positive organisms. It is under light microscope. Now, these are the organisms. I would say this is which I am taking is that is the organism which is causing the gas gangrene. And these organisms are, I would say previously, the human and animal feces and surfaces in the room where there is oxygenation is there because they are anaerobic organisms, unwashed hands, soil, water, food, and best media is the meat one. These are the species which I have said. There is this one that I am talking about. There is gas gangrene, the Clostridium Perfinges. Maybe some rare species are involved, like the Clostridium novae and septicum and sodali and stereoticum. Sodali is important in septic erosions. And Clostridium Perfinges can also cause the gastroenteritis. Now these species of Clostridium are the obligate anaerobe. They are definitely bacillus and gram-positive bacilli. Basically, they are rod-shaped, and in structure is concerned, and already said that there is Schaffer-Fulton stain, 0.5%. Malachite green in water can be used. It from there is anthrax, which is very near organism to it, which is also flourishing in the same area as Clostridium. So it can be differentiated by this Schaffer-Fulton stain. Now, this is another thing which I have told you previously also. These organisms are obligate anaerobic growth. They have got catalase negative, and they are therefore the oxygen is not there, the oxidase negative. And it can be detected by the PCRs. This is under the low microscope. So this is the technique which I described it previously, which I have described it in front of you, and the tetanus which I have described previously next day, yesterday I think, and I described it as far as concerned, because a lot of actually lectures have been recorded, so I missed whether I have told you or not. So this is what I have actually, I think I have already discussed it. Now this is the infection which I want to discuss. That is Clostridium perfringens. Now Perfinges is Clostridium welchii, or Bacillus welchii, is a gram-positive, rod-shaped, elegant, anaerobic. It forms spores, yes, spore forming. It is catalase negative, oxidase negative organism. The genus is Clostridium. I will say the genus, it is a big one. More than 100, 110, 120 species under this heading. And the humans, for the humans, there are 4 or 5 are important. Rather than 7 are important. Under light microscope, you see this rod-shaped, gram-positive bacteria. And the source of infection as Clostridium perfringens is concerned, it is ever present in nature. They said that they are present since 5,000 or 10,000 years ago. Or some researchers said that when the earth was created, they were present in the soil. It is a normal component of decaying vegetation. Any vegetation which is decaying, it is a normal component. For the commensal is there, because you have to remove this the decayed part of the vegetation. They help in the removal of the vegetation. Marine sediment is concerned, intestinal tract of the humans, again they are present. Clostridium is different type of species. And the vertebrate insects in the soil. It has the shortest reported regeneration time of any organism, because 6.3 times in thioglycolate medium. Glycolate medium, it will 6.3 minutes doubling time. Generation time is, or doubling time is 6.3 minutes only. So its power of growth is very, very high in comparison with the other organisms. The clinical microbiology of Clostridium perfringens is concerned is stable G plus C content. Around 27 to 28% has a genome size of about 3.5 MB. And Clostridium perfringens strains have since been made available on NCBI genomes database for the scientific research community. This is the research center. The genomic research has revealed surprisingly high diversity in Clostridium when genome is concerned. As our whole genome is concerned, there is high diversity. Only 12.6% core genes identified as the most divergent gram-positive bacteria reported. On the same component, they may be weakly gram-positive or even gram-negative are possible. That means that. Nevertheless, the 16S RNA regions in between the Clostridium perfringens strains are found to be highly conserved. That is called sequence identity are more than 99.1%. So they can be detected on the specific media. Their growth rate is very high. Motility, they are motile organisms or not motile. Although they do not have an flagella, CD, etc., Clostridium perfringens bacteria are able to glide. You know what is glide? Gliding across the surfaces. They move by gliding because their bodies are lined with the filaments from end to end, filamentous type of structure. Under the high microscopic field, you find there are filaments in the body. The hypermetal variants such as SM101 are often found arising on the edges of the colony's agar plates. Or video microscopy of their gliding movements suggests that they form the long thin filaments that allow them to move rapidly like bacteria with flagella. The segmentation moves one segment to the other segment to the other segment. In this fashion, they glide. The gliding bacteria, they are gliding bacteria lacking flagella or other moving tools like cilia, microcilia, and pseudopodias. No. Only segmentation. Segmentation, they are actually forming and then moving ahead. It means the displacement is due to the filament they are forming. And their doubling time is 6.3 minutes only. So this is important in this sequence, which I am telling you about this clinical microbiology is concerned. So they are gram positive, and they are anaerobic. They are segmented structures. They are normal. They have no flagella, cilia, or pseudopodias. But the mortality is good. They are gliding type of bacteria. They are gram-positive bacteria, and they are the gliding bacteria. So they glide without mortality. So mutations in the cell division genes is the common feature of the hypermotile strains. Strains which are hypermotile, mutations are seen. Against it, there is a problem of to found it as far as the active immunization is concerned, it's difficult to attain. The culture, you see these colonies, they are moving on this, and they are high microscopy. You see filamentous filaments. This is the filament. 1, 2, 3, 4, 5. Again 5, 6, 7, 8. These filaments will be detached, and the other filaments go out, and out. And they glide like this. So you can use the media. I already mentioned about the media. And the media in this condition, as I already said, that their media is specific to differentiate the other gram-positive organisms, specifically the Bacillus anthrax is concerned. To use this malachite stain, green stain, to differentiate with the Bacillus anthracis is concerned. In a culture media, if you want to identify it. As I said, the agar is used with different type of the organic material you can use as a culture media. Now, clinical presentations are as follows. First, I want to talk about this food poisoning. When you are using the word food poisoning, it means that it is the gastroenteritis or gastroenterocolitis related with the food. It means that you are able to demonstrate the gastroenteritis related with food intake. This is called food poisoning. Now, food poisoning in the humans is caused by the type A strains able to produce these CPE for the Clostridium perfringens enterotoxin, that is. This enterotoxin is a polypeptide of the 35.5 kDa that accumulate in the beginning of the sporulation, because they are spore forming and excreted to the media, where it lysates at the end of the sporulation. It is coated by the CPE gene, that is enterotoxin, present in less than 5% of type A strains. Now, in the United Kingdom and United States, America is concerned, how the Britain is concerned, where a lot of research has been done. The Clostridium perfringens are the third most common cause of the foodborne illness, food poisoning. With poorly prepared meat and poultry or food properly prepared, but left to stand too long, the main culprits in harboring the bacteria. Well, they are like to eat, the meat, and they like to eat meat in different fashions. So the slaughterhouses, they're doing their work. They cut into pieces, refrigerated, and then the supermarkets, they are selling it. After you will buy this, or those people who are buying from this, they make the food, or the prepared foods are there, and this is the ideal time when the Clostridium perfringens can enter into this organic material. If you have heated it, fried it more than 100 degree, 105 to 200 degree Fahrenheit, temperature more than 200 or 300 rather, they are killed 165 degree. About 165 degree Fahrenheit there, but you can do up to 200 degree Fahrenheit. The deeply fried or the very highly heated, you have to cook it like this. If you leave this food and go for the refrigeration, retake again the food again, and then heat it in the microwave, for example. Now, again, you have to reheat that thing. It's okay, no, fine, because the lethal dose is not appearing, but it is appearing afterward. If you are reheated again and again, then it will become a problem. It will become a problem. It is ideal part now for the growth of the Clostridium perfringens. In our country, where this Clostridium perfringens and enterotoxin are able to penetrate it, think it about. Are the super stores which are preparing, or multinational companies who are preparing this food, because they are checkups are going on repeatedly. They are checking. From America, the people are coming. The franchise companies like McDonald's and KFC are concerned, and others as well. They're coming after 1 month or 2 months and check everything. They take the sample and then they go into the lab. They send it to the lab. Either the Clostridium perfringens or any other organism can be cultivated from the refrigeration or not. The first sample is there. Second sample was the sample which was cooked properly by the company, fast food company, famous companies. Again they will culture it. They will double check. Because if they want to continue this and their popularity, they have to do this. Otherwise, the other fast food stores or shops, they are not actually following the instructions given. They are using 3 months old beef or mutton or chicken. They don't bother about it. But you are taking the burger or what you are like, pizzas or like this thing, which contains this organic material, like anything, the fish or whatever it is. And it is 2 months old, 3 months, expired actually. For them it is expired, but they do not throw it away. And they prepare it for you. You cook it and delicious. After going inside, the incubation period is about 6 to 24 hours. Or rather, I would say it is 10 to 12 hours. It's better to use half a day, you will feel the nausea, vomiting, and then diarrhea. Now this diarrhea is the diarrhea you develop very commonly in our country. Remember, they are spore-forming organisms, they are motile organisms, they are resistant organisms to the environment. Remember this thing. That the food poisoning depends. If you are evaluating the food poisoning, then the question should be asked: what you have taken in 1 week before or 2 weeks before, start from 2 weeks before or 3 weeks before. Have you ever visited these the wedding ceremonies or any ceremonies or these food centers, the junk food centers, or these centers, fast food centers, and there are other centers as well, restaurants? What you have taken. Second question is what you have taken from there. Third question is that when you have taken it, how many days after this you feel the abdominal cramp, vomiting, diarrhea, etc. And does it persist now? If she said yes, it means that the organic material which you have engulfed is still in the intestine, and although the coliform are fighting against it, but they are not able to it because the enterotoxin is liberated now. And enterotoxin definitely, if it is entered into the blood, it is causing the toxemia. The Clostridium food poisoning also due to another disease known as the enteritis necroticans, that is Clostridium necrotizing enteritis. It necrotizes the small intestine. It is also known as the pig bill. Pig bill, this is caused by the Clostridium Perfinges type C, necrotizing enteritis. And usually it is fatal because of enterotoxemia and because of the septicemia. Large numbers of the corpuscle intestines and secrete exotoxin. This is enterotoxin, exotoxin. This exotoxin causes the necrosis of the intestine, varying levels of the hemorrhaging and perforation of the intestine. Inflammation also occurs in the secretion of the jejunum and the midsection of the small intestine. It is limited disease to the small intestine, it means that. And in some cases there may be possibility of the ulcerations. Definitely if it is entered into the body, it will lead to the same process, the bacteremia, and then septicemia, and then pyemia, the SIRS, and sepsis, and the septic shock, and MODS, so on and so forth. Another important thing which I want to discuss is that the parasite Ascaris, it is producing the trypsin inhibitor, Ascaris lumbricoides. So Ascaris lumbricoides that contain large amount of the trypsin inhibitors, sweet potatoes, areas prone to infection of the parasite Ascaris, which also a trypsin inhibitor. The sweet potato is also a trypsin inhibitor. So in these cases, the sporadic feeds the meat, that is the long period of the protein deficient diet, they are trypsin inhibitors. So these are the again predisposing factors, although they have not taken the diet containing the organic material. This disease is contracted in population living in these areas: Africa, Central America, South America, and Asia, Southeast Asia, and in Arabic, Saudi Arabia and around it. It is possibility for the growth of this organism in sweet potatoes and the people who've got Ascaris lumbricoides long-home infestations. So they are prone to, they are predisposing factors. Now again, in many cases, the Clostridium perfringens food poisoning, the main subclinical antibodies to the exotoxin are common among the population. Repeated infection going on, you are taking, I'm thinking of one child, or आपने देखा होगा, the very poor children, they are eating the food you have thrown it in the wedding ceremony. And the wedding ceremony में जो है ना, they discarded the food and they put in the bucket. They have taken that food. They should develop this poisoning, definitely. Or other poisoning, but no, very rarely they come to us with the food poisoning is concerned. Because they have taken subclinically the exotoxin, and exotoxin prone to develop the antibodies in these population. That is why these people are very, very hard people. Even the highest powerful countries, world powers, they are failed to conquer the Afghanistan. They are highly resistant people. They can engulf anything. And with the grace of Allah, antibodies are formed, they are safe from the disease. I have never seen one person who become corona positive. And in Afghanistan also, it is very rarely the people belonging to the original one which are called. They do not have this infection. And actually it is in Karachi, there is also an Afghan basti where the people, a lot of people are living, they have no corona infection at all. Or any other infection like this, which is Clostridium perfringens food poisoning. Why? Because the subclinical infection they are taking. आपने डिस्कार्ड कर दिया, उन्होंने खा लिया. They have taken it. And in this way, indirectly, they are modifying or enhancing their immunity against these infections. Despite its potential danger, the Clostridium perfringens is used as a leavening agent in salt rising bread. जो ब्रेड बनाते हैं तो इसमें जो कमेंजल के तौर पर इस्तेमाल होगा ये. The leavening agent के तौर पर इस्तेमाल they are using it. The baking process is thought to reduce the bacterial contamination. High temperature होता है वो मार देती है. उससे जो काम लेना होता है they have done it. Salt rising bread जिसमें जो लोग बनाते हैं, because उससे food poisoning नहीं होती, because they act like the commensals rather, and they are facilitating the process of the baking. Now the clinical feature, another one is the gas gangrene. It is a dangerous thing. That is, it will cause the gas gangrene. Of the gas gangrene symptoms, any localized infection. First of all, there is cellulitis, then there will be destruction of the muscles, compartment syndrome. Then it is entering into the blood, and they will cause the SIRS, systemic response syndrome, to going to the sepsis and septic shock. Also involving the liver, and the jaundice, the biliary stasis occur in this condition. Any septicemia can cause jaundice. A strain of the Clostridium perfringens might be implicated in multiple sclerosis, and this is the Clostridium immunity actually. Nascent pattern 3 lesions. And these are tested in the mice. The toxin made by a rare strain of the Clostridium perfringens cause the multiple sclerosis-like lesion and damaging the brain. And another work had identified that this strain of Clostridium perfringens in human with MS. I have already said that at one time, say after 50 years or more than that now, from onwards, 2080, you can say something near to the 2000, 70 around it. There are possibilities every disease may be related with the microbiological agent. Initiating agent will be found to be microbiological agent. So like for example, in this, multiple sclerosis, pattern 3 is related with a factor which is Clostridium Perfinges they are secreting. And they have proved it in the mice, and they are also found in the human also. If a microbiological initiating agent is found, you can definitely manage the case very easily, because a lot of antimicrobials are available. You can do it. Even for the cellularity, the blister formations, all the necrosis occurs, and in the necrosis part, you find there is capitis here. You touch it, there are capitations, because the gas forming organ it is and it is called the gas gangrene. If it all this part is necrosed, ischemic necrosis with the putrefaction is called gas gangrene or gangrene. If the gas is produced, it is called gas gangrene, or dry gangrene, or wet gangrene. So it is ischemic necrosis with the putrefaction of the bacteria. It is called gangrene. This is a very dangerous situation, because the patient, it may be amputated from here to here, yes. But if it is untreated as a septicemia, then it will go through all processes of the septic shock and MORS and ARDS and DIC. The patient can be saved, but it is very, very high, it is called high mortality. That is why if it is limited to here or here, the limb should be amputated. Amputation should be done as early as possible. Diagnosis is a colonies, of course. You have to culture it. Double zone of hemolysis, irregular margins colonies are there, but there is a Nagler reaction in which the suspected organism is cultured on the egg yolk media plate, Naglers, that is. If the Nagler reaction is positive, what is the Nagler reaction? The rise of the Clostridium inoculation here, and here. Okay, now there are segmentation occur and crossing here. Opacification occurs due to the lecithinase activity of the alpha toxin have caused lecithinase activity. So opacification occur here. The less oxygen is there. And the second part and the third part also. And the fourth part, when you have actually induced the anti-alpha toxin one on half of the plate here in this area, there will be no antitoxin on the another side of the plate. Lecithinase will not be able to opacify it. If you culture like this, it means that only one organism can cause it, and that is called Clostridium perfringens. That is called the Nagler reaction. Why I am talking about it, egg yolk media, why I am talking about Nagler reaction. Remember, some examiners, they have already done the clinical microbiology, and afterward they become consultants. And there are many, many examples of it. The foreign examiner, they like to discuss on the clinical microbiology. Clinical microbiology is a part of internal medicine. I'll say, always remember, it is part of it. 1/3, or rather more than that, is covered by the clinical microbiology. The clinical microbiology should know something about it, Nagler's reaction, can differentiate it. A streak of the suspect, organism is placed, I have already told you the toxin, the alpha toxin indicating and the uninhibited lecithinase activity, which is penetrating activity, is high, that is why. And gas is formed. If the individual has more than 10^16 spores of the bacteria per gram of the stool, then the illness is diagnosed as a Clostridium perfringens food poisoning. Now this is at least count your stool is culture. You can do it from the blood also. and there are other medias are there, or other tools are there, gas, liquid chromatography, etc., etc. They can be diagnosed. You have to send the tool. The agri reaction, they do this, the other culture count of this bacteria is more than 10 to 6, 10 to power 6 spores. It was said that you are suffering from this poisoning, and the food which you have taken from which place where, that restaurant will be sealed if it is proved. कोई कराता वराता नहीं है, लेकिन किसी को फूड पॉइजनिंग होगी तो फिर उसने अपने आप को जिम्मेदार करार दे दिया, दूसरे लोगों के लिए कोई फ़ायदा नहीं पहुँचाया। अगर उस टूल को करा लेता और वो जो सैंपल था जो जहाँ से उसे खाया था वो भी उसे वहाँ से भी कल्चर कर लेता, दोनों जो थे मिमिक कर लेते, तो दैट इज डायग्नोसिस कि ये सोर्स ऑफ इन्फेक्शन है, इसने ये प्रोड्यूस किया था, ये रेस्टोरेंट जो है अनहाइजीनिक है, ये क्या कर रहा है कि रेफ्रिजरेशन इनकी पुअर है, 2-2, 3-3 महीने के पुराने जो होते हैं ऑर्गेनिक मटेरियल वो उसने प्रिजर्व किया हुआ है अपने पास, उससे वो बना रहे हैं। आप देखेंगे कुछ रेस्टोरेंट ऐसे हैं जहाँ पे ये जो मिलती है, बिरयानी भी मिलती है, आप खा लेते हैं, कुछ भी नहीं होता, कि उनकी बिक जाती है, वो रखते ही नहीं है उसे, और इसी वजह से अभी तक वो कामयाब है। 40 साल हो गए उनको, अभी तक कोई भी उन पर केस नहीं कर सका, इसलिए कि वो बनाते हैं, फरोख्त कर देते हैं उसी वक्त, और जो बच जाती है वो गरीबों में बांट देते हैं। इसके लिए तो आपको प्रूफ डायरेक्टली ही करना पड़ेगा। दूसरा तरीका ये है कि आप जो है ना PCR से इसको प्रूफ करें या एलाइजा से प्रूफ करें, ये भी पॉसिबल हो सकता है, उससे भी ये डायग्नोज हो जाएगा। आसान तरीका ये है कि आप कल्चर कर लें, कल्चर मीडिया पे जो है वो स्पोर को डिटेक्ट कर लें नेगुलर रिएक्शन करके। ट्रीटमेंट इसका क्या है? ऑफकोर्स ये है कि एक तो सर्जिकल मेजर है, वो तो करना ही करना है, डिब्राइमेंट है, सर्जिकल डिब्राइमेंट है, इवन इम्पुटेशन शुड बी डन। If it definitely there is no pulses behind it, you have to impute it, or there are signs of toxicity develop. अभी Clostridium जो है, these are positive, positive, they are very very sensitive against the beta-lactam antibiotics. All beta-lactams, whether penicillin, cephalosporins, monobactams, carbapenems, even the fusidic acids, or even nicomycins, and now macrolides 3rd generation and fluoroquinolone 3rd generation, they are also effective. They are ICU patients, and IV administration should be given for the antibiotic to save this patient from the septicemic shock. Prevention is better than cure. यहाँ पे आके जो है ना हम जैसे कंट्री वाले मारे जाते हैं। फूड इंस्पेक्टर्स होते हैं आपके पास, इन आवर कंट्री देयर आर फूड इंस्पेक्टर्स, दे आर नॉट डूइंग ड्यूटीज ऑनेस्टली, दे टेक भत्तास फ्रॉम दी रेस्टोरेंट्स एंड गो अवे। मरते हैं तो लोग मरें, दे आर नॉट इंटरेस्टेड। लेफ्ट ओवर फूड शुड बी रेफ्रिजरेटेड टू अ टेम्परेचर बिलो 4 डिग्री। तो आपने खाना खाया था ना, जैसे आप लेके आए थे फिश विद फ्राइड फिश विद फ्राइज लाए थे या चिप्स लाए थे, प्रिपरेशन के बाद जो है ना उसको क्या करना है, आपने 4 डिग्री सेंटीग्रेड विद इन 2 आवर्स ऑफ प्रिपरेशन आपने इसको जमा दिया। लार्ज पॉट्स ऑफ फूड सच एज सूप और स्ट्यू विद द मीट शुड बी डिवाइडेड इनटू स्मॉल क्वांटिटीज। बड़े बड़े भगोने जो आप लोग जमाते हैं किसी भी सालन वालन के, वो ना जमाएं, थोड़े थोड़ा पार्ट दे के जमाएं। अगर जमाना है आपने दोबारा इस्तेमाल करना है, और अगर दोबारा इस्तेमाल हो गया तो फिर वापस नहीं रखा जाएगा। अगर हमने फिर रीहीट किया दोबारा, तो फिर इसके साथ चांसेस हैं अगेन, या आपकी 165 डिग्री अचीव नहीं किया आपने गोल फ़ारेनहाइट का, उसमें भी ऐसे ही होगा कि ये जो है ना रीहीटिंग जो है बल्कि 200 डिग्री फ़ारेनहाइट पे करनी चाहिए बिफोर सर्विंग। और रूल ऑफ थंब इज दैट इफ द फूड टेस्ट, अब असल बात ये इंसान बड़ा सेंसिटिव है, स्मेल तो लुक्स डिफरेंट फ्रॉम व्हाट इज सपोज्ड। अगर आप खा रहे हैं और खाते खाते जो है ना आपको लगेगा कि इसका टेस्ट मुख्तलिफ है, ये स्मेल इसमें से सही नहीं आ रही, और वो इसकी जो शेप भी है वो डिफरेंट हो गई है मॉर्फोलॉजिकली। इन सबसे अक्लमंद तरीन वो लोग जानवर जिनको आप तस्वीर नहीं भी कर सकते, वो कैट की फैमिलीज हैं, वो डॉग की फैमिलीज हैं। जो ये जो एनिमल्स हैं इनकी हिस्स बड़ी ज़बरदस्त है, क्योंकि ये ईट करते हैं मीट को, तो ये सूंघ सांगते हैं, बहुत देर तक उसे सूंघेगा। आप देखिएगा, बिल्ले को डाल के देखें, ये चीज़ जो चीज़ आप डिस्कार्ड करने वाले थे, वो बिल्ले को आप डाल के देखें, वो बहुत अच्छी तरह से चेक करेगा उसको। चेक करने के बाद खाएगा, ऐसे नहीं खाएगा। उसको ज़रा भी लगा कि इसमें कोई प्रॉब्लम है तो वो छोड़ देगा, नहीं खाएगा। इसलिए डॉग भी इसी तरह करता है कि बोन, उसको मरगूब की तरह वो बोन को अच्छी तरह से सूंघता सांगता है, फिर उसे चबाता है। इंसान को इतना होना चाहिए कि टेस्ट चेंज हो गया है, कोई बात नहीं यार, हो गया तो क्या फर्क पड़ता है, अच्छी स्मेल नहीं आ रही, खा लेते हैं लोग फिर भी। या मॉर्फोलॉजिकली ये दाल नुमा चीज़ थी, अब ये सालन नुमा हो गई, फिर भी खा गए। तो ये तो मतलब आपकी बेवकूफ़ी ने ह्यूमन जो है ना वो ज़्यादा हिमाकत कर रहा है ना इन टेकिंग दिस। लेकिन जानवर बड़ा समझदार है, वो उसको नहीं खाएगा। तो ये मल्टीप्लिकेशन जो है इसका 12 डिग्री सेंटीग्रेड टू 50 डिग्री सेंटीग्रेड के दरमियान अच्छे मल्टीप्लाई होता है। ऊपर जाएगा इससे अगर आप ले जाते हैं तो फिर मल्टीप्लिकेशन कम हो जाएगी, या 12 डिग्री से नीचे आए, 4 डिग्री से नीचे आए आप, या ये रेफ्रिजरेशन पॉइंट माइनस में आ जाएंगे तो फिर भी मल्टीप्लाई नहीं कर पाएगा। लेकिन अगर आप बड़े बड़े भगोने अगर रखते हैं, जैसे रखते हैं लोग रेफ्रिजरेशन में, तो उसके अंदर जो है ना ये मल्टीप्लाई हो जाएगा। उसके असल में बात ये, उतने टेम्परेचर जो है ना वो अचीव नहीं कर पाएगा बिलो 0 डिग्री से नीचे की बात कर रहा हूँ, कर नहीं पाएगा ना। तो ये इसके ऑर्गेनिज्म के बारे में दो खसूसियात हैं क्लॉस्ट्रिडियम परफेंजेस बेल का। एक तो ये फूड पॉइजनिंग कॉज करता है, दूसरी गैस गैंग्रीन फॉर्म करता है। दोनों का मैनेजमेंट ज़ाहिर है कि सर्जिकल डिप्लॉयमेंट में फूड पॉइजनिंग अगर आपको देना है तो ज़ाहिर है आपको बैलेंसेस पूरे रखने चाहिए पेशेंट के, इलेक्ट्रोलाइट बैलेंस है, फ्लूइड बैलेंस है, ग्राम पॉजिटिव के खिलाफ आपको इस्तेमाल करनी है। ये देखिएगा, आइडियल ड्रग इसमें इस्तेमाल होती हैं, जो आजकल इस्तेमाल होती हैं वो 3rd जनरेशन कैफलोस्पोरिन को देते हैं लोग, और इसके अलावा जो है वो क्लोरोनिक एसिड एमोक्सिसिलिन भी देते हैं, वो भी देते हैं इसमें। और अगर ज़्यादा कोई टॉक्सेमिक है तो उसमें तो ओरल डी देते ही नहीं, उसमें सब इंटरवेनस थेरेपी होती है। अब आप चाहें तो इसके बाद वाले लेना चाहें तो आप इस्तेमाल कर सकते हैं। मैक्रोलाइट या 3rd जनरेशन है, या फ्लूरिक्विनोन 3rd जनरेशन है, वो इसको भी बड़ी ज़बरदस्त कवरेज देती है। तो इसलिए जो है ना ये चीज़ आनी चाहिए। मतलब ये कि हमने हमारा काम तो बताना है, हमने बता दिया अब आपको, इसको याद करना अब आपका काम है, समझना या रिपीट करना। ये तो वीडियो है, इसको दोबारा देख लीजिए, 1 दफ़ा 2 दफ़ा देख के आप किताब में पढ़ लें। अब आप किताब में पढ़ेंगे तो जितनी तफसील मैंने बताई वो नहीं मिलेगी। और आप अगर कोई फेलोशिप एग्जामिनेशन दे रहे हैं या हायर एग्जामिनेशन दे रहे हैं ख़ास तौर पे, तो वहाँ ये मारे जाएंगे आप। इसलिए आप इसको समझ के पढ़ें, और जो कुछ मशवरे दिए मैंने, प्रिवेंटिव भी मशवरे दे दिए आपको, और उसके भी मशवरे दे दिए हैं, दोनों मशवरे दे दिए हैं, मैनेजमेंट वाइज भी दे दिए हैं, हर चीज़ बता दी है, इसमें कोई क्लू नहीं छोड़ा है, सब कुछ बता दिया। वमा अलैना इल्लल बलाग। अस्सलाम वालेकुम वरहमतुल्लाहि वबरकातुहू। So we will continue with another Clostridium species and tomorrow inshallah.